Dive down to the surface of a sequencing flow cell, and you enter a highly engineered microscopic landscape. Across a single glass slide, millions of isolated chemical reactions are firing continuously. A single Illumina run generates up to 50 billion bases of data while maintaining a global accuracy rate of 99.9%. The engine driving this output is a technology known as sequencing by synthesis, or SBS. Yet for all its processing power, SBS hardware hits a strict physical wall. The machine cannot read a continuous strand of DNA longer than roughly 300 bases. To understand why the process collapses at cycle 300, we have to look closely at the molecular mechanics. The exact same chemistry that ensures near-perfect accuracy also ensures the system will eventually break down. This diagram illustrates the sequencing environment at the molecular level. We have a single DNA template strand, a polymerase enzyme, and a flooded environment containing all 4 nucleotides: adenine, cytosine, guanine, and thymine. To increase sequencing speed, Illumina introduces all 4 nucleotides simultaneously. When the correct nucleotide binds to the template strand, a lock drops into place, physically blocking any other bases from attaching. This gating mechanism works because each free-floating nucleotide carries 2 precise modifications. On one side, it has a fluorescent dye attached. On the other side, the 3' end is capped with an azetamethyl blocking group. Once the base is locked in, a camera records the specific color emitted by the dye: green for adenine, red for cytosine, and so on. After the image is captured, a reducing agent called TSEP washes over the strand. This chemical cleaves both the fluorescent tag and the blocking group in one step. The strand is now open, and the cycle repeats. Older pyrosequencing methods, like the 454 system, washed nucleotides over the DNA one type at a time, without a blocking group. If a strand had a sequence of 5 adenines in a row, all 5 would attach at once, causing a bright flash that was difficult to count accurately, a phenomenon known as a homopolymer error. By forcing the polymerase to pause after every single base, reversible termination eliminates homopolymer errors. But there is a catch. The light emitted by one single fluorophore molecule is too dim for a camera to detect. Zooming back out to the surface of the flow cell, we face a hardware limitation. To capture an optical signal, the camera requires millions of identical molecules firing the exact same color in the exact same location at the exact same time. This diagram demonstrates the solution: bridge amplification. A single fragment of DNA anchored to the flow cell bends over, its free end binding to a complementary adapter nearby. A polymerase enzyme then moves along this arch, synthesizing a complementary strand. The double-stranded bridge is chemically severed at the top, leaving 2 identical copies that stand upright. The original strand is washed away, leaving the reverse strand anchored and ready to bend over again. This looping physical reaction is repeated roughly 1,000 times, confined entirely to one microscopic spot on the glass slide. The result is a monoclonal cluster. You now have roughly 1 million identical copies of the exact same DNA fragment tethered in a tightly grouped lawn. Now, when the sequencing cycle begins and the reversible terminators attach, 1 million identical fluorophores flash their respective colors in unison. This creates a dense optical dot bright enough for the sequencing camera to read. If the chemical blocking prevents errors and the cloning provides a clear optical signal, why does the entire process shut down after 300 cycles? This cross-section view shows 10 cloned strands within a cluster. Because chemical reactions are probabilistic, enzymes occasionally make mistakes. During a cycle, a polymerase might completely miss an incorporation. One strand fails to grab its nucleotide. This specific strand is now lagging exactly 1 cycle behind the rest of the cluster. In the next round, it will display the color for cycle 50, while its neighbors are displaying the color for cycle 51. This is known as phasing. The opposite error is prephasing. Sometimes the TSEP fails to cleanly cleave the blocking group, or a defective terminator slips through. A strand jumps ahead, incorporating 2 bases at once and flashing a color meant for the future. A single dysynchronized strand doesn't ruin a 1 million copy cluster. But over 50, 150, and eventually cycle 300, thousands of strands fall out of sync. Some are lagging, some are jumping ahead. The 300 base limit stems from a physical collapse of the signal-to-noise ratio. The camera can no longer distinguish the true color from the overlapping flashes of desynchronized strands. The optical signal simply blurs into an unreadable gray. Mapping short 150 base reads across long, repetitive genomic sequences creates a specific structural ambiguity. If a sequence repeats identically across the genome, a single short read lacks the unique landmarks required to anchor it to a specific coordinate. This animation shows the primary hardware workaround, paired-end sequencing. Instead of trying to read through the entire fragment, the sequencer targets the edges. The machine synthesizes 150 bases in one direction. Then, the newly built strand is stripped away. The original strand folds over into a bridge again. The polymerase copies it, and the forward strand is washed off. The reverse strand is now exposed, allowing the sequencer to run another 150 bases from the exact opposite end. This is read 2. The machine does not sequence the middle of the fragment, but because the laboratory protocol strictly controls the total length of the initial DNA fragment, the insert size, we know exactly how far apart those two short reads are. Paired-end sequencing provides geographic context. Knowing the distance between the two reads allows bioinformaticians to uniquely anchor the fragment, identifying structural variants without ever physically reading the distance in between. This limitation also clarifies Illumina's advertised long-read platforms, which boast lengths of 5 to 7 kilobases. The flow cell is not actually reading a continuous 7,000 base physical molecule. Instead, laboratory prep uses tagmentation. Transposase enzymes fragment a long DNA strand, while simultaneously inserting bar-coded adapter landmarks along the breaks. The flow cell sequences these chopped fragments in standard 150 base bursts. Software algorithms then look at the barcodes and computationally stitch the short pieces back together to create a synthetic long read. The architecture of an Illumina flow cell dictates exactly what it can and cannot do. It sacrifices the ability to read long physical molecules in exchange for a rigidly controlled chemical gating system. It is this exact trade-off, forcing millions of amplified clones to pause step by step, that delivers the high-volume throughput and accuracy that continues to drive modern genomics.